Hello, and welcome to SeedWorld's Strategy webinar. Today, we're going to explore the importance of in-furrow technologies, the latest science, and how they impact seedling development. As many of you know, my name is Julie Deering, and I'm the editor of SeedWorld. Today, I have the fortune of serving as your moderator. First, I'd like to take a minute to thank our sponsors, BASF, Bayer, and Syngenta for their support. I do want to let you know that this webinar is being recorded and will be made available on seedworld.com following the event. As we go along, if you have any questions or comments, please share them in the chat box and we'll do our best to address them at the end during the Q&A session. Today, our speakers are, Brian Keel of West Central, Adrian Jewell of Bayer, and Karen Schmidt of BASF. And we're going to be discussing the role and evolution of in-furrow technologies, how in-furrow technologies interact with seed treatment and seedling development, and the latest science and technologies available. After the presentation, we'll open it up to questions. As I previously mentioned, if you have any questions, just type them into the chat box. So our first speaker is going to be Brian Keel. Brian serves as West Central Director of Product Development. He joined West Central in 2000 to start its product development department, where he focused primarily on adjuvants, nutrients, and crop protection products. In this role, he was instrumental in bringing the first in-furrow fertilizer solution for iron deficiency chlorosis, soy green, to growers. He also led West Central's efforts for developing its complete line of in-furrow fertilizers that are currently on the market. Brian is active in professional societies, having served on committees such as the Advanced Crop Advisors Workshop Committee and is director of the North Central Weed Science Society. He earned his master's degree in weed science from North Dakota State University, and his areas of expertise include herbicides, insecticides, fungicides, adjuvants, and agricultural fertilizers. Brian, the floor is yours.
Thank you. Thank you, Julie. A few years ago, West Central, in cooperation with, West Central, in cooperation with BASF, FMC, Dow AgroSciences, and New Farm, founded an initiative that we referred to as LIFT. LIFT stands for Leaders of In-Furrow Technology. This initiative basically revolved around ensuring that plants were given every opportunity to establish themselves and thrive in what is usually a harsh growing environment early in the season. It involves the efficient use of early season inputs that are critical to maintaining and enhancing early season growth. These inputs could involve fertilizers used in furrow, they could involve nitrogen stabilizers, they could involve seed treatments applied to the seed, and insecticides and fungicides applied in furrow to help protect the plant from environmental or some other harsh stress, such as diseases or insects. At West Central, we've been observing this trend towards in-furrow for quite some time. In-furrow puts the best products in the best place at the best time to ensure that the best early season growth and development of that plant takes place so that plant can maximize its yield later in the season. Today, it's estimated that about 25% of growers utilize some sort of in-furrow application, and at the growing rate of the trend, we expect that to increase to about 50% of the growers by 2020 at the present adoption rate. In-furrow applications are actually synergistic with broadcast foliar applications of fertilizers and plant protection products, and they absolutely contribute to maximizing yield of the overall plant growth. What I want to talk about is I want to focus on starter fertilizer. One of the keys of nutrient uptake in plants is proper placement of your nutrients so that they're accessible for the plant, and making sure that those nutrients, just because you put them in the soil, we also want those nutrients to be available to them. Here, good stuff. For starters, you need nutrients to get that plant up to a growing start. You can utilize both macro and micronutrients and place them right next to the developing seedling so that those early season roots have easy access to those plants or to those nutrients. We meet the plant's nutritional needs right from the start with an overall balanced plant nutrition system. And this gets our plant off to a good start. That improves early season growth, uniform growth, and we get off to a great start so that we can improve overall plant health. This leads to a stronger root system that not only benefits the plant from being able to pull up the starter fertilizer that is in the root zone, but it also benefits the plant that root system can then expand out of the root zone and extract other nutrients from the soil profile. One of the technologies that West Central has developed is a technology that involves a chelating agent that is very unique to the agricultural marketplace. This chelating agent is utilized in a number of products that West Central offers for sale to growers. Some of these products are very critical from the standpoint of applying in a complete fertilizer package, such as Levisol and Levisol DFC, whereas Levisol is a liquid product that can be added to liquid fertilizers to help make those fertilizers more available. Levisol DFC is applied to granular products as an impregnation type of an application, and that allows those fertilizers to be more available to the plant. And then we have products such as soybean and soybean granular that have very specific functions where they help make iron more available so that when soybeans are raised in situations where iron chlorosis is a problem, that the iron is more readily taken up by the plant. And then starter fertilizers that are complete packages such as Redline and Soy Shot that can be used in corn and soybeans to offer up both macro and micronutrients to be more available to the growing plant. This chelating agent, again, is the most stable chelating agent that is utilized across multiple soil types and multiple pH ranges. And this chelating agent functions by attaching itself to micronutrients that are in the soil and preventing them from being tied up in the soil and preventing them from interacting with other nutrients in the soil. A good example of this is what happens when zinc and phosphate interact. Basically, you have zinc that is a positively charged micronutrient, and you have phosphate that is a negatively charged macronutrient. Two form a bond, and they form zinc phosphate. When they do that, they are not available for plant uptake. When we utilize one of the products that utilizes the ortho-EDDHA chelating agent, that attaches itself to the micronutrient that is in the soil, such as zinc, or other micronutrients, manganese, copper, or iron, and that prevents the zinc and the phosphate from forming a bond where each individually can be now taken up in the plant. An example of this is in the soil extraction study that we have conducted throughout multiple soil types. I have an example on your screen. We have 3 different soil types, one from Texas, one from North Dakota, and one from Minnesota. And if you look at the gray bar, which is hard to see because it's basically non-existent, when we try to extract manganese out of that soil, you can see that we are not able to extract any manganese out of the soil in a normal condition. When we add our chelating agent to the soil, you can see that we can extract quite a bit more manganese out of that soil, and it differs based on the soil that we are using. When we look at a standard soil test of these 3 soil types, we actually show that there is manganese in the soil, and it actually is reflected by the amount of manganese we are able to extract when we add the ortho-EDDHA chelating agent to that soil. Concurrently, when we take a look at phosphorus that was in those same soils, when we add the ortho-EDDHA to the soils, we are able to extract more phosphorus out of the soil. And again, the chelating agent does not interact with phosphorus at all. It interacts with micronutrients that are in the soil, such as zinc, such as iron, such as manganese, and it prevents them from interacting with the phosphorus, and therefore the phosphorus becomes more available to the plant. An example of a product that we have available is soygreen, as I mentioned before. And soygreen contains the highest percentage of chelated iron with ortho-EDDHA in the industry. It is the highest percentage of water-soluble and stable iron that is available to growers today. It's been on the market for about 14 years. Soygreen used in soybean trials has resulted in about an 8.2 bushel per acre advantage versus not utilizing soygreen. But in some cases, we actually get as high as 40 bushel an acre or more when we utilize it in harsh iron chlorosis situations. The slide that you have in front of you looks very busy, but basically it's extremely simple. When you look at the blue bar, that is soybeans grown in a soil that has iron chlorosis, and all of these soybeans were growing in the same field, but they're all different varieties. And if you look at the white number at the base of those soybeans, that is the iron chlorosis rating for the tolerance of that soybean to iron chlorosis. And if you have a 1, it's very tolerant to iron chlorosis. If you have a 3, it's not as tolerant to iron chlorosis. And as you can see, that based on soybean variety, we can get a tremendous yield response from utilizing soybean in this field because it allows the soybeans to grow normally and uptake iron and not be exposed to the iron chlorosis problem. You can see our largest yield responses are in situations where we have aggressive varieties that are probably higher yielding varieties, but they don't have very good tolerance to iron chlorosis. The other thing that I have mentioned is the impact of this technology on phosphorus. There are a number of factors: pH, amount of clay, organic matter, whether or not the soil is weathered, that can impact phosphorus and phosphorus's availability to the plant. Now, you're always going to have some sort of phosphorus in your soil, or phosphorus tie up in your soil, but in certain situations, you'll have much less available phosphorus than in others. The chelating agent that we utilize in soybean, Levosol, Redline, and soy shot does allow phosphorus to be more available to the plant. An example of this is a trial that was conducted at Kansas State University, and I apologize, some of the numbers are off of the bars, but essentially when you compare the use of an untreated check versus a starter fertilizer, in this trial we resulted in a 17 bushel per acre advantage. When we added Levosol to that same starter fertilizer that freed up nutrients to the plant, we then increased our yield by 9 bushel to the acre. Now, I don't just show you this just to show you a yield response. The other thing that we looked at was the amount of phosphorus that was in that plant. When we evaluated the yearly phosphorus percentage, it correlated perfectly with the yield response, where when we used the starter fertilizer, where we had phosphorus available to that early season growing plant, we did increase our phosphorus at the end of the growing season in the ear leaf by about 14%. And that explained partially why we got a yield response. And the same thing happened when we added Levosol to that starter fertilizer, where we increased phosphorus in the ear leaf by about 10%, which again correlated to our yield response. That's just a snapshot of some of the products and the technology that West Central has in what we call our Levosol family. Again, this includes Levosol, Levosol DFC, which can be added to either liquid or dry fertilizers, Soygreen, which can be applied in a liquid system, Soygreen granular, which can be applied in a dry air seeder situation, both specifically for iron chlorosis in soybeans, Redline, which is a complete package of macronutrients and micronutrients that can be used in corn, and Soy Shot that can be utilized as a starter fertilizer in soybeans. Thank you for your time today, and with that, I'm going to turn it back to you, Julie.
Thank you, Brian. That's interesting to see. Is it safe to say that like the ortho EDDHA increases the sustainability of production because you're making use of the nutrients that are already in the soil instead of having to add more?
The ortho EDDHA makes a more efficient use of both fertilizers that are, or nutrients that are already in the soil, as well as nutrients that you're applying to the soil. Correct. Okay,
great. Well, thank you very much. I appreciate that. Next up, we have Adrian Jewell. Adrian, let's see here. There we go. Adrian is a product development manager with Bayer. His responsibilities include soil applied biologics and insecticides and digital farming development. He has worked with Bayer for 4 years, spending the first 3 with the biologics group in West Sacramento, California, leading the foliar entomology team and an insecticide discovery project team. Adrian's current role brought him back to the East Coast, and he now assesses the potential of his products through field trials across the country. Adrian is passionate about agriculture and the potential of biologics to bring greater sustainability. He balances his love of research with spending time with his wife and 3 young children. And I have to apologize, the last bullet on my slide is incorrect. He earned his doctorate, but it's in forest entomology from North Carolina State University. So we'll have to get that updated. Adrian's going to talk to us about in-furrow technologies and how they interact with seed treatments, especially from the standpoint of biologicals. And Adrian, I'll let you take it from here.
Okay, thank you, Julie. So here at Bayer, at the moment, we don't have a lot of products that are applied in furrow and corn, but we are developing some. And so I'm going to focus on how we're going about that development process and how to think about the different inputs that lead to an early plant establishment. So as we start the season off here, most of you who are farmers out there have probably already chosen your genetics and your seed, and you have a seed treatment package that's already in there. And then upcoming, as you go to actually put the seed into the soil, you'll have an opportunity to also use different in-furrow inputs. As Brian discussed, the leading input in furrow at the moment is the liquid fertilizers, and where I'm developing are things that go usually along with that in-furrow fertilizer, so either an insecticide or a biological. Some of the reasons that we go in the furrow is that in addition to the commercial seed treatment, you can really fine-tune your needs for exactly what's happening locally on your farm. You really then also get to focus in a larger area of the root zone, so as the roots come out of the seed and start to move through the soil, they'll still be in that in-furrow space. You can have more volume in furrow, so seed treatment you tend to be limited in the amount of things that you can put on the seed. Because the seed already has an integrated solution package including fungicides, insecticides, and then possibly some microbes that allow for greater plant growth. And when you go in furrow, you can then put more material in there, which is quite important for certain inputs. Currently, most of the products are individual products that will just be one AI, but there are also some emerging combos. And really, again, the fertilizer tends to lead with the insecticide being the second choice there. Now, mostly those pop-up starter fertilizers tend to be a little further to the north, especially important when you put your seed in cool soil where it might have to really be able to take advantage of any warmth that's coming in. And then where you're putting in that insecticide is where you know that you have greater insect pressure, and also where you might be trying to add an additional mode of action to protect from developing resistance to either traits or other modes of action. Similarly, a fungicide could be added in furrow if you have a really high fungicide, a plant pathogen infection in your soil, or use less tillage. And where I'm going to focus today is plant health biologicals. So these are things that are actually living organisms that you're going to put into that root zone. And I'm going to talk about both exactly what those are and then how you think about putting a living organism into that space. So the one we have in the market right now is Serenade ASO, and so I'm going to use that as our example, but we're also developing other technologies. And these biologics are generally characterized by their active ingredient, which is the living organism. And that's characterized by the number of living cells or spores that you're actually receiving in the product. These spores, the living ones, and so each living spore can then reproduce and form a colony if you put it in a nutrient medium. And so that's why they're called colony-forming units, or CFUs. So that's capturing the number of living organisms that you're able to put into the field. So you can see there's a little star at the bottom. I will try to mouse over that so you all can see where I'm pointing. And so if you look here, it has a minimum of 1 × 10⁹ CFUs per gram, and so that's how much you're actually getting in terms of those spores. And then also on that label, it tells you what percentage are those spores of the total volume of the product. So the product, there's also, as you're growing that bacteria, you have all the things that you put into the fermentation broth in order to get it to grow. So you have all of those, as well as things that the bacteria produces while it grows, in addition to those final spores that you end up with. And so within that, you're getting the living organism, you're getting the spores, and you're getting things produced. And one of the common things you might be able to think about to put that in context is if you drink beer, the alcohol there is produced during an anaerobic fermentation. And so that's one of the products that we like in beer. And we don't have to, and with the fermentation of other microbial products, we get different types of metabolites produced. So now to differentiate the biologicals from small molecules, when you think about a small molecule, you're getting one active ingredient in the package. So if you buy Poncho and you're putting Poncho on the seed, what you're getting is clothianidin. That is the active ingredient in Poncho. And so there's the molecular structure of that, goes on the seed, and then it's both uptaken into the plant. It provides a zone of protection around the seed, but you're really just looking at that one molecule. When we think about the biological, on the other hand, you're getting the living organism itself. So this would be the spore of the bacillus. In this case, it's known as QST713, is how that is patented, kind of that organism's name. And it produces a number of different compounds. And so some of them are lipopeptides, and that's this group here at the top. And these have these hydrophobic tails, and that allows them to pierce into plant pathogens and kill the plant pathogen. There are also other families of chemistry that also help to defend the plant and to kill plant pathogens. And then there are others still that help to promote plant growth. And so these are compounds like 2,3-butanediol and indolacetic acid. Both of these are plant hormones and help the plant grow. Now we're going to take and look at how the microbe actually looks as it's growing in the system. So the top series of pictures that you see are the plant root growing in a clean nutrient media. So this is not growing in soil, because in soil you'd have other microbes that would also help to colonize the root, but we're just looking at this kind of in a lab setting to understand what it looks like. And so normally here's the clean root, the top left picture, moving across a fluorescence microscopy, so being able to zoom in a bit further, and there we can see the root hairs as well. And then finally a scanning microscopy image where we can really look at the outer surface of that root in great detail. On the bottom, you can see what happens when the root is colonized by the serenade microbes. And so from light microscopy, you can see it looks a bit fatter. And this is the microbe growing around that root and forming a biofilm in the rhizosphere, or that region around the root where the root is putting various compounds that the microbe can grow on into that environment. And so these root exudates that the plant is putting out actually help microbes colonize that space, because the plant wants to grow with microbes in the soil. In the second picture, we can zoom closer, and you can see while the biofilm has formed around the root hairs are still able to penetrate through that biofilm and access the soil space, but it is forming a barrier to protect from other sorts of pathogen attack. And then finally, if we zoom all the way in, we can see the individual microbial cells growing in that biofilm, along with exopolysaccharides, which kind of help to form a more concrete film there, as opposed to just individual organisms. So now I want to talk a little bit about the benefits you get from this interaction. So this is a beneficial interaction between the plant and the microbe. Both benefit. So the microbe has this area where it can grow around the plant root, and then the plant also gets to grow better and has these additional elements that help the plant. So you get increased both root and plant growth through plant hormones. It increases soil nutrient availability. And so we just had Brian talking about how you can get some starter fertilizer elements that help to maintain the availability of both inputs as well as elements that are already in the soil. And microbes also help the root interact with that area around the root in order to access more of some of those micro and macronutrients. It's through compounds that are given off by this rhizosphere colonization. So clearly, as you're going in with different inputs, you can think about what certain inputs give versus others, and how you're kind of building a better plant establishment situation. So then you have that biofilm where you have a physical barrier to pathogen attack, and in some cases nematode attack. And it also primes plant immunity to repel root and foliar pathogens. So when you have a microbe growing in that space, communicating with the plant ends up being more ready to be able to defend itself against pathogens as well. And now I'm going to move into talking about compatibility. And so first of all, as we start to test our biologicals, we make sure they're compatible with Bayer seed treatments. So Bayer, as you all know, has the Poncho Votivo seed treatment out on the market. And since that also has a living microorganism, Votivo, we make sure that any other microbe we put in the system gives compatible and additive benefits to what you already get with having Votivo in the system. And so microbes as living organisms in a living system have to find their own niche and then compete for that space. And so in that competition, they have to fight each other off. And human antibiotics are an excellent example of how we utilize that. And so we also can use similar types of chemistry in plant pathogen protection and have to think about them and how microbes play with each other and make sure we choose microbes that play well together. What you see on the right is two white colonies, and those are different colonies of serenade. And then around the edge of the plate, you have a plant pathogen trying to grow in. That's the fungus botrytis. And you can see there's a clear zone outside of the serenade colonies where the botrytis is unable to grow. And that's because this is called a zone of inhibition. And there, the chemicals produced by the serenade microbe as it's growing prevent that plant pathogen from growing into that space. And we see similar interactions in certain soils with certain pathogens where they are unable to invade that rhizosphere space when it's inhabited by the serenade microbe. And so not only is there the protection element, but also now as we're putting multiple microbes in the system, we have to make sure they grow together as opposed to fighting each other off. So for example, with Votivo, we've shown that you do get that additive beneficial interaction. And in addition to that sort of biological interaction, it's also important to make sure you have the physical compatibility. So when you take and you formulate this biological, there's a lot of stuff in that fermentation mix that you're putting into the system, and you want to make sure that's physically compatible with your fertilizer as well, so that you don't end up clogging up your lines that you're using to apply those liquid in-furrow inputs. And so as we're developing new products for this in-furrow space, we're making sure that we're very compatible with what's on the seed, and then also compatible and able to be flexibly used with various in-furrow inputs that also may be chosen from other people, like, as we just heard, the liquid fertilizer element. And so lastly, as you're making the decisions around in-furrow inputs, make sure that the solutions are both physically and biologically compatible. And then also think about what the pressures are on your specific farm. What other targets might you need to hit that aren't already covered by either your germplasm or your seed treatment? Also think about how your in-furrow solutions then are building you the best full program for early plant establishment. And make sure that you're investing in liquid in-furrow application systems that let you flexibly pick different in-furrow solutions as they become available and with different properties. And then just a little bit of legal wording here about the things that we've said here as possible forward-looking statements. And I want to thank all of you for your attention and pass it back to Julie to introduce the next speaker.
Great, thank you very much, Adrian. And our next speaker is Karen Schmidt. Karen has recently had a position change and now serves as U.S. Field Biology Group Leader for BASF. Before taking on this role, she served as Technical Marketing Manager with BASF Crop Protection and focused on headline AMP, fungicide, and corn. Here, she was responsible for technical positioning, training, and product use recommendations. She was also responsible for helping growers and retailers use BASF crop protection products effectively and sustainably to maximize yields and plant health benefits. Karen has also worked as a technical service representative supporting Michigan and Ohio and as a project leader in the Herbicide Development Research and Development Group. She earned a master's degree and doctorate in weed science from North Carolina State University and a bachelor's degree in agri-science from Michigan State University. Karen, it's all yours.
Good afternoon, everyone. Thanks, Julie. Really appreciate the opportunity to be here today to share sort of the BASF perspective of the role of in-furrow solutions in corn production. So for the remainder of the webinar, there we go, a few topics that I wanted to touch on today were basically grower insights and what has changed and what's really driving the interest and need for in-furrow solutions for corn production. Some of the additional grower needs today and why in-furrow now. And then lastly, I'll wrap up with some in-furrow options from BASF, and from our standpoint, we basically focus on fungicide and fungicide-insecticide solutions. So by way of background, as we think about in-furrow and some of the drivers behind the interest and increase in in-furrow usage beyond liquid fertilizer, we know that U.S. corn producers are getting larger every year, and those really large producers are growing at a rate of about 8% annually. So with that, labor and time are really at a premium, and growers continue to plant earlier and really push the envelope on planting corn, in essence, to cover more ground and also to maximize that yield potential from those earlier planting dates. But earlier planting really can put greater stress on germinating and emerging corn crops. So whether that's cold and wet soil conditions or additional disease pressure or insect pressure. So looking at planting early, a 10-year analysis that was conducted by the USDA showed that there was any range of suitable planting days over a 10-year period from about April 30th to May 20. So this was really focused on the central corn belt. But there were, over this time period, as few as 4 days and as many as 19 days. So the range can be all over the place in terms of available days for planting. But we know as growers get to planting in the spring, they can cover a lot of ground very quickly. One comment is in 2014, 28 million acres were planted in just 1 week. But with all of that opportunity to plant faster and plant earlier, again, the seed's not always going into ideal soil planting conditions. So what kind of impact does that have? What it means is that the corn seed is being put into conditions of possibly cooler and wetter soils oftentimes, and that there's potentially a greater chance of disease pressure. Now, most commercial seed would have a fungicide seed treatment, but it's going to be limited in the amount of protection that it can provide. It's going to offer protection really in the beginning of the life cycle of that seed. And lastly, many trials have been conducted, but in our own research, we found that 6 out of 34 plants, when we withheld 6 out of 34 plants by 5 to 7 days, yield was reduced by 12 to 18 bushels per acre. So if you think about a planting population of 34,000, if 6,000 of those are delayed by any period of time, yield potential can be significantly reduced. So getting that crop off to a good start is critical. Now that's in kind of the agronomic drivers behind in-furrow, additional in-furrow interest, but there's also economic drivers. If you look kind of historically at, say, the mid-70s to 2006, the cost of production per bushel was essentially flat. And crop prices were also lower back in those days, but there was an increase in yield annually at about 2% per year. So that yield increase was necessary and really helped to increase income. Now, in 2007 to 2013, when commodity prices were much higher, yield was a luxury, but the crop commodity prices often outset or overcame the cost of production per bushel. Now, of course, we're back in more modest commodity price era, and so really the only way to reduce cost of production per bushel is to increase yield. And that doesn't necessarily mean reducing inputs, but it really means investing in proven technologies that increase yield potential. And that's where a lot of the reasons why the in-furrow has become of such interest. Now, related to the in-furrow fungicide acre, it's really a new segment in crop protection, combining the liquid fertilizer that the previous speakers have spoken about, with or without insecticides, to provide the best opportunity for the crop for more rapid and uniform emergence and increased seedling vigor. So this chart just shows the optimum planting date. This is for central Illinois, but these dates can be adjusted for any geography. Basically, there's an optimal planting time, and if you go too early, it's a little less than optimal. If you go beyond that, it becomes suboptimal, and of course planting later, yield potential is decreased significantly. And so growers trying to cover all their ground and trying to optimize that yield potential window or planting earlier. But if you look at that orange bar, that's showing kind of the inverse relationship of soil temperature, where getting the crop in the ground at the right date may not always coincide with the soil temperatures being ready for planting or being optimal for planting. So there's a trade-off that has to occur in getting timely planting and also having ideal conditions. And so any number of technologies that can protect against those stresses at early planting are important. So why in-furrow solutions now? Here we're looking at a photo of an untreated side of a field compared to a Xanthion in-furrow fungicide treated side of a field. This is from 2016 growing season. And just visually you can see the significant reduction in number of plants where there was no fungicide applied in the furrow. Now, Xanthion is a fungicide technology from BASF that combines the active ingredient in headline fungicide and pairs it with a biofungicide. So it's a dual mode of action fungicide product for soil-borne diseases as well as plant health benefits. So Xanthion protects against soil-borne diseases and then also provides growth efficiency and stress tolerance benefits that are well documented. And you can see that very visually here, that whatever held those plants back in the untreated, that yield potential is lost. Any yield potential that's lost at the establishment time cannot be made up for later in the growing season. So getting that establishment period optimized is critical. And of course, the crop has to be maintained and managed throughout the rest of the growing season in order to maximize that yield potential. So looking at in-furrow fungicide solutions and fungicide insecticide solutions from BASF, we have two products in the marketplace currently. And I mentioned the Xanthion fungicide, and again, that's a dual mode of action chemical and biofungicide combination product. And then we also have Manticor LFR fungicide insecticide. And this is where we would recommend that if additional corn rootworm control is needed in addition to the fungicide component of Xanthion. So Manticor combines the active ingredient in headline fungicide, and it pairs it with the active ingredient in Capture LFR insecticide. So really targeting the additional control of corn rootworm as well as secondary insect pest. So we'll start with what Xanthion fungicide does and how it, and I want to just talk about how it works or complements well with the fungicide seed treatments and how the chemical and biological components can all work together. So basically what we have is the seed is planted and it has the fungicide seed treatment, which is really that first line of defense against seed-borne diseases as well as early infection of soil-borne diseases. And then the chemical component, so that headline component of Xanthion fungicide, protects against soil-borne pathogens, and especially as that seedling starts to develop, and it really extends that zone of protection beyond just what the seed treatment can offer. Next, the biofungicide component within Xanthion starts to grow and develop along that root surface. So that's depicted here as that kind of green shield along that root zone. And that, as Adrian mentioned previously, produces antifungal metabolites that control the soil-borne pathogens, but it also has it also physically excludes those pathogens from getting into the root system of the corn seedling. And then over time, of course, that seed treatment is going to start to dissipate because you can only get so high of a level of active ingredient load, but the chemical and biological component continue to provide protection in the soil against diseases, but then they also impart those plant health benefits that I described, helping under tough conditions of cold, cooler and wetter soils. And then lastly, as the chemical component starts to dissipate in the soil over time, that biological component, because it's a living organism, does continue to grow in abundance over time, and that extends the length of the residual control. So it's between the seed treatment, the chemical control, and the biological control, you have a full protection against seed-borne, soil-borne diseases, and then also those plant health benefits. And then transitioning then from Xanthion to Manticor, and again, this would be the product of choice where corn rootworm is a concern. Of course, corn rootworm doing most of its damage in the larval stage, and it's been tagged the billion-dollar pest because it's estimated that the costs associated with controlling corn rootworm total $1 billion annually for U.S. corn growers. And with Manticor LFR, what we've done is partnered with FMC to utilize their competencies in LFR liquid fertilizer ready formulation technology and their insecticide technology with our competencies in our fungicide, plant health fungicide technology. So this is a ready to use liquid fertilizer ready product. And just to show the mixability, on the left side you see a just 1034 liquid fertilizer, and then you see in the middle the components that are contained within Manticor as individual formulations, EC formulations, and then on the right is the ready-to-use liquid fertilizer ready formulation, where it's all equally suspended within that liquid fertilizer. And then just to show some of our field trial results, where we're looking here at corn rootworm root ratings, which is a 0 to 3 scale, where the 0 is no root feeding and 3 is 3 sets of nodal roots that have complete feeding by corn rootworm. And the Manticor LFR provided excellent protection there against corn rootworm ratings, against corn rootworm feeding, compared to the untreated. And then we also had commercial standards in there, and there was very comparable level of control. So again, this would be for growers wanting that disease control and plant health benefits of the headline fungicide, but also wanting the additional corn rootworm protection. So I'll just wrap up my portion and think about, a grower in their lifetime has about 40 opportunities to establish a crop. And really they're striving to make every opportunity their best, wanting to make each year better than the year before, and then certainly wanting to maximize yield potential as we're in leaner economic times. So an in-fertile application can protect the grower's investment well beyond those normal seed treatments and extend that zone of protection. The fungicides from BASF Xanthan or Manticor LFR provide the developing seedling more rapid and uniform emergence to really provide a quicker start to maximize yield potential. And it does so by protecting the corn seeds and seedlings in conditions of disease pressure and stressful soil conditions like colder, wetter soils. And again, just trying to get that seedling and crop off to the best start and the best chance for survival, and really trying to optimize that yield potential. And so with that, I'll close and I will hand it back to Julie for Q&A.
Great, thank you very much, Karen. So with that, we're going to go ahead and open it up to questions from our viewers. So we ask that if you have any questions, please go ahead and type those in the chat box, and I will ask them of our speakers. And so, Karen, one of the first questions that came in is for you. When you were showing the slide that had Xanthan, the test there that was performed, was that an SGR base or a fully untreated seed?
So the comparison photo, was it, what was the question? Was it can you repeat that?
Was it with an SGR base or on a fully untreated seed?
Gotcha. That would have been, that side-by-side comparison would have been seed treated with just a commercial seed line, and the only difference, so both sides would have had that, and then the only difference would have been that addition of the Xanthan applied in furrow.
Okay, great, thank you. And then the next question here is for Adrian, or it popped up when Adrian was giving his presentation, and it's a bit long, so I'll do my best. It says, regarding organic production, federal standards and mandates require strong oxidizers and copper-containing compounds for fertilizing organic crops. Both of these type chemical spray initiate browning responses in plants through the ROS and upregulate the translation of polyphenol oxidase and polyphenol lanine lyase and peroxidase, all of which initiate rapid browning. Are there any chelating agents available to sequester hydrogen peroxide and copper to prevent stressful conditions in organic crops? So that tripped me up there, but Adrian, I'll let you go ahead and take a stab at it.
Given that it was asking about the chelating agents, I was kind of hoping Brian would take that one, but I could try to say something about biologicals in that if you'd like.
Sure, why don't we go ahead and pass it to Brian?
Okay. Essentially, the chelating agent that we have in the products that I described, the ortho-EDDHA, it does chelate copper, and so it would make copper more available to the plant, regardless of whether it was copper application to the soil or whether there was copper already in the soil. However, I believe the question surrounded organic crops. We have not done a lot of work with organic crops. We have more so used this product in traditional crop production, and so we would not have any type of OMRI certification or anything like that with any of our products that I mentioned.
Thanks, Brian. And then the second part of that question is, off of the microbial inoculation, are there any available biologicals that are symbiotic and can tolerate salty soils, or even process salty soils to allow for less saline conditions?
That's a great question. So I know that we did a little bit of work a while back looking at Serenade in rice systems, and I believe we saw some benefit of Serenade in rice. As you increase the saline, you'd still have more tolerance in rice. I'm not exactly sure what the mode of action was for that, and we haven't done any of that work in corn and kind of salinated areas. But I know in rice we were able to get some benefit from adding the microbial. And a lot of times you see kind of a greater root mass, so that also might just help the plant tolerate that salt a bit. But that's all I can say about it.
Great, thanks, Adrian. The next question that we had is for Karen. You mentioned an 8% annual growth in large growers. How do you define large grower in terms of acres?
So I saw that show up in the chat box, and I was trying to find my source for that. And let's see. I can't come up with it. It's a source from Successful Farming. I can't find the actual delineation what the cutoff is for large farm size in terms of acreage. So I apologize for that. I can't come up with it at the moment.
Okay. We'll try to figure out what that is from information or data from Successful Farming, and then let the person interested in that know. And,
okay, and I can dig it up too as well. I just can't put my finger on it right at the moment.
Sure. No, that'd be great, Karen, if you can do that. Thank you.
No problem. Sure.
And then, are any of these biologicals available for sorghum and for export to Southeast Asia?
Okay, that's a great question. So I believe they can be used on sorghum. We are labeled in sorghum, say for our Serenade ASO. The bigger question tends to be, how do the specific microbes interact with the specific plant? I actually tested Serenade in sorghum last year, and the results kind of on average did not show a huge positive effect. We are going to test it some more this year, because there were one site that looked very good, one site with no effects, and one site that looked negative. So it wasn't that much data. Now, for the export side of the question, the biologicals that we have, the Serenade is exempt from tolerance, and therefore there aren't regulated residues that you have to worry about. So it's very safe. It's a probiotic for chickens as well, the same microbe, so very, very safe. So export-wise, you'd be good to try it, but I don't yet have the data showing where it would bring you the sufficient benefit in sorghum, but we are working on it.
Great, thanks, Adrian. And this next question goes to Karen. How do your technologies interact with yield enhancers, PGRs, or microbiomes, and are there positive or negative effects?
So we basically, we don't test a lot of those combinations ourselves, but we have seen them in field trials in terms of side-by-side comparisons. And in many times we've seen a lot of instances where there is an incremental benefit with those combinations, kind of a stepwise progression, and we've seen that numerous times with some of the other in-furrow products.
Okay. And then the one last question I have for Brian is that when was Lift, or when did that come to be, and kind of what was the driver behind that, or how did that get started?
Sure. It was about 2.5 years ago where Lift came to fruition. And what it really was a collaborative effort amongst the companies that I mentioned, where all of us were involved with applications that were made early in the season that benefited the developing seedling. And working together, we could actually reap the benefits of the synergy of all of the products, whether it be a plant health-based product used in furrow, an insecticide used in furrow, and our chelating agent technology used in furrow, where we could get that plant off to a good start so we can maximize yield potential.
Okay, great. Well, I think that wraps up our time for today. So I want to take a moment to thank our speakers, Brian, Adrian, and Karen, for their time and sharing their expertise. I hope that you've found this information valuable. And again, I want to thank our sponsors, BASF, Bayer, and Syngenta, for their support. And on behalf of my team here at SeedWorld, working behind the scenes, I want to thank them for helping to make this a successful event. Again, this presentation will be made available online at seedworld.com. Thank you for your attention, and I hope you have a great day.